thanks for the nice introduction. Today I will be talking about the human bone and bone marrow chip. And actually, as Katrin already mentioned, this has been a group effort. So for everyone in the audience, this is not a single woman thing. You have to have a pretty dedicated team around you with different projects. And yeah, I hope I can present some of that work today. And yeah, I'm excited for the discussion later. And I really want my colleagues to talk about their topics. I won't keep them from answering questions. So today I would like to talk about the following things with you: the origins of our cells and tissue that we use on the bone and bone marrow chip, then what kind of quality assessment or pre-testing we do and how important seeding is. I will also present the first stress test that we did in a clinical setting with our bone and bone marrow chip. And then I will go on talking about native tissue. I will show you pictures you will understand later and how that's handy when you do basal research. Then we will talk about osteoclasts, the bone resorbing cells, very special cells, as you will learn, hopefully. And then a little bit about immune cells and the soluble environment and our future projects or the outlook. I would like to mention that I will show some images that might be disturbing to some of the audience. It's basically human tissue, femur head, if you're a bit into anatomy. So if you don't deal with these things or blood very well, you will find in the top right corner a little red flag, and that means the next slide will contain such images. Actually, for the first one, it's going to be 2 slides in a row, so I will try to remember to tell you when the pictures are out of your site, so you can continue looking at the slides. So generally important cell types for us in the bone and bone marrow are the osteoblasts. They're bone forming cells, and the bone resorbing cells, the osteoclasts. And our skeleton is actually renewed all the time and actually in a balance. So it's not that one day you make a lot of bone and then the next day you lose a lot of bone. Maybe it happens that way, but usually we think it's 1-to-1. So the bone you form, you also resorb, and then the skeleton is actually renewed over time and balanced out. If you have some issues, you can develop osteoporosis or osteopetrosis, which is actually more rare, and it's usually dysfunctional osteoclast, but it will come up later again. Also, the mesenchymal stroma cells are important to us. So they are basically part of the connective tissue. They can differentiate into the osteogenic direction, so they can make bone, so to say. And then we have the bone marrow mononuclear cells that also includes the hematopoietic stem cells, but also adult immune cells and developing immune cells, and also the monocytes. So monocytes, they are more commonly known in their further developmental status, which is the macrophages, the gobbling cells, but they can actually give rise to the osteoclasts. So they're also in there, but the general word for that is the bone marrow mononuclear cells. So starting, so I have these little subsections, so you can rest your eyes and you can breathe and you can maybe digest all this input a little bit better. So first, I would like to tell you where we get our native bone and bone marrow samples from. Please appreciate the red flag. We have actually very dedicated colleagues that form a core unit called tissue harvesting, because they get samples from the clinic. So if there's a surgery, in our case, if you get a hip implant, then bone is naturally removed during surgery, and patients can graciously donate that to our cause. So we have a team that actually organizes all of that and provides us with primary samples, which they also quality check. The most interesting tissue for us, or the most cell-rich tissue or liquid in this case, is the so-called bone marrow aspirate. So some of you might know it's the liquid part of the bone marrow. It's usually also used in transplantation. And the spongy bone, so the bone substance. But sometimes we would like to say, when we're lucky, we also get the femur head. So that's, that goes into the joint as depicted in the picture. So maybe I should use my laser pointer. So here, And that will also be shown later how that can be utilized. Okay, so we have the bone marrow aspirate, and from that we then isolate, or the core unit actually, or whoever needs those cells, we can isolate the osteoblasts, so the bone-forming cells, the mesenchymal stromal cells, and the bone marrow mononuclear cells that I talked to you about earlier. What we can't is isolate osteoclasts. So the bone-forming cells cannot be removed from tissue. They stick to the bone like gum, and yeah, they don't really like to be isolated from primary tissue. So lots of our people in the lab then quality check those cells. So we want to know if they actually make bone, right? So we do these in regular in vitro assays, so 2D. We stain the matrix and we look at all of the different aspects. So do they form matrix? Is it actually going into the right, in the osteogenic direction? And with that, we do alizarin red staining, who's familiar with this thing, and also look in alkaline phosphatase, which is actually responsible for accumulating phosphate to then make new bone. And we also do flow cytometry of the immune cells that are present within the bone marrow, because actually we're pretty heterogeneous. That wasn't really important in the past decades, but like about 5 or maybe 10 years ago, people realized that bone marrow is actually immunologically pretty interesting, and I'm very interested in it. So we always do like a subset characterization of blood and bone marrow immune cells, and want to see how it differs, especially how it differs also between patients. So just to show you that we're really doing it, this is the lymphocyte cells in the blood and the bone marrow. We also look at early B cells. I will not use the laser pointer again because I'm a little bit afraid of it, of having a technical issue again. So we also look at B cells. I'm very interested in B cells, and it's also a quality check if we have early B cells in the bone marrow because they're not present in the blood. But of course, the heterogeneity you can see here, depending on the age or what kind of life that person had, they have different levels of subsets in their bone marrow, which is interesting on its own. When it comes to the more adult B cells, there's actually no significant difference when you compare blood to bone marrow. We also do the same for T cells, and here it's interesting, or it's just nice to show. It has been described in literature before that the ratio from CD4 to CD8, so the T helper cells and the cytotoxic T cells, they're actually inverse. When you look at blood, it's 60-40 that way, and then the other way around in the bone marrow. So we could just confirm that, because another good thing to do when you work with chip or with native tissue is always, does it add up with the data that has been published in humans prior to your own work? So the next part is we were looking into how important it is to mix cell types and how they influence one another. I mean, most of you that did something with cell biology know that cell-to-cell contact and all of those things are quite important, how cells will behave later on. So my colleague Annie, actually, she did all of the work. This is just a small excerpt of that. She tested different media and then different cell compositions. And we could actually see, or Annie actually showed, that using 70%, 75% mesenchymal stromal cells and 25% osteoblasts, so bone-forming cells, if you mix them together, they make really nice matrix, which later on then can become bone, so tissue and then bone. So this was actually really nice, and it's just to kind of illustrate that you always have to think in different layers. So in this case, Annie showed that this was a nice mixture that you can then use in 3D, because it would be sad if you do all the work putting the stuff in 3D if it doesn't already work in 2D. So now, but this is called on a chip. So I proudly present the chip. We actually use Tissue's 2-organ chip. It's a company based in Berlin. And the 2-organ chip, along with other chip models, are commercially available from Tissues. And it works something like this. It's a microscopic slide. And you have 2 circuits embedded onto that microscopic slide, and they're independent from one another. So they have two wells that are connected, and then you have laminar flow. You can perfuse that. You can see that here when it's plugged in, but you have two separate circuits on one chip. And for us, we add bone scaffold in one, and the other one serves as a medium reservoir. Because that's pretty cool, because you can just unscrew one lid, do a media change without disrupting the bone that is in the other chamber. So schematically, it looks something like this. Very nice in BioRender. We actually work completely xeno-free, so no animal-derived material used here. That took a lot of time because, you know, you have so much, you have so many options on the market, and we actually use human serum that we add to our different media. It really depends on what research questions you have, but when you start a project, you basically just mix media and see what works, because sometimes too much glucose causes one cell to thrive and the other one just, you know, die. But in general, here again, I introduced all of them to you before. We use human decellularized bone. So this is actually from the biobank at the Charité. There are little biopsies that fit nicely into a 96 well plate. And we then equip them with osteoblasts, mesenchymal stromal cells, the bone marrow nuclear cells. And maybe you remember that within this population of bone marrow mononuclear cells, we also have the monocytes that are differentiating into osteoclasts. When I first started, we just thought, we just add some stuff, and then the monocytes will just naturally differentiate into osteoclasts. They don't. We needed another person to find that out, but you will hear about all of that later. So this is how the setup looks like right now. And it's nicer to show this how it looks in real life. So here are some pictures from the lab. You can see the bone scaffolds here, the decellularized biopsy. So we punch that out. And then we have again, the osteoblast, the mesenchymal stromal cells, the bone marrow mononuclear cells. We put all of that on the bone. We plug it in, and then we have a nice flow. The media just perfuses throughout the whole system. And then we just basically monitor what it's doing, kind of like a Tamagotchi. So again, we add all of those cells, and then we let that run for about 35 to 42 days. It really depends what research you're doing, but we do say that you need about 3 weeks in order to have bone matrix. So here on day 28, so when you start, you have like, this is a light microscopy picture because I told you it's a microscopy slide. So you're actually able to check your little scaffold within the tissues chip during its run with a light microscope. So yeah, you can see that it's pretty empty, so to say. Of course, there are lots of cell there, but I haven't made any matrix. And then after 3 weeks, here it's day 28, we see a typical bone structure. It's called reticular connective tissue. It's a typical collagen. It's found in bone. And then later on, you really see that this chip is packed with cells, and it's making all kinds of matrix and tissue. So it's actually really fascinating every time you're watching the scaffold growing, not bigger, but denser and denser. To just show you some readout parameters that we use in our lab, just briefly that you've heard it, we monitor bone cell activity and viability, especially at the beginning in 2D. We check for matrix deposition because we want to know our cells really making bone in the end. During the run and also afterwards, we look for bone formation and resorption. We actually do that with really fancy nano and micro CT and ELISAs. We check for the structural integrity with microscopy and synchrotron analysis. This is like super high resolution X-ray, so to say, in a simplified way, but it's really cool. You actually get to go work with a particle accelerator, not in CERN, but in Grenoble. So it's really, really nice, and you can get good images. I will show some pictures later. We monitor cytotoxicity with LDH release. It's an enzyme. I show that later as well. Morphology, immunohistochemistry, We look at the heterogeneity of the immune cells, especially, and then again, soluble environment like cytokine arrays and MSD, if you know this platform. Okay, so now I have given you a lot of information, but now I really want to show what we then did when we came up with, when we got everything running. So it was the first real life stress test. And we always want to try to benchmark our system to the closest available reality. And for us, that's actually patients. So we have the luxury to talk to the people in the clinic. We are all part of the orthopedic department, and we ask them, what kind of problems do you have, you know, during surgery or whatever, and how can we help? So in this case, when you get a hip or knee implant, it has been improving the quality of life for many people, but some people actually suffer from something called aseptic loosening. So for some reason, they just lose bone around their implant, and at some point, it just gets so loose that it needs to be replaced. And you cannot forever replace knee and hip implants. At some point, you can't. The bone is just too fragile. So our question was, can we expose our chip system to the same metal cocktail that is present in patients? And do we see the same reactions as our colleagues in the clinic describe as well? So here is a nice biorender schematic again. So we use clinically relevant concentration of chromium and cobalt. They're usually present in hip and knee implants. And we just basically dumped that on the chip and wanted to see what happens. Our first readout was the LDH. LDH is an enzyme that is released from dying cells. Of course, cells are dying all the time, but if the LDH is increasing in a very massive way, then you probably can see that something's wrong. And this is just a commercially available assay, so we can monitor that. So we first started off to see what cell numbers is actually how many cells do die from so you have your assay, and then you maybe want to know how many cells, what does it mean? Like, I have my assay, but how many cells actually died during the process? So we had a colleague running some assays, so we basically knew what kind of cell numbers we're talking when we measure LDH values within our chip system. And for chromium and cobalt, we could actually see that over time, the LDH concentration was increasing. And when we looked in flow cytometry, we could actually see that the monocytes, you remember the monocytes, that they were actually decreasing, which is interesting because the monocytes and also macrophages are very well described in this process where you lose bone when you have an implant. So this was a nice finding because it's actually matching what our colleagues in the clinic are also observing. When we looked in with immunohistochemistry on day 28, we could actually see that chromium and cobalt was destroying the bone matrix. I think it's pretty impressive how you can see that this is destroyed tissue. When we did an element mapping, so here you can see in red, it's actually calcium, so that means bone, and then it's a bit small. I know, I'm sorry for that, but just to get the point across, we could actually monitor how chromium and cobalt was distributing itself in the bone, in our bone scaffold. And then here, this little fuzzy thing, that's actually a region where we would see osteolysis. So if bone gets resorbed, it kind of gets like all mushy and the bone is just not stable anymore. So this was really cool because we also had patient samples with the same pathology, and it looked the same as in our chip. So that was pretty cool. Cool enough to publish it. Actually, a really nice paper came out during the pandemic in close collaboration with the orthopedic surgery department. And that was the first success when we were, just trying to go back and forth from bench to bedside and back. So now I showed you like all the complex ways how we equip the chip. But we actually also came up with an idea that maybe we can just use native material for initial research questions. So actually, my colleague Martin has really optimized this way of work, which is pretty cool. And we came up with the idea, or Martin actually came up with the idea to punch out little biopsies, the same cylinder as you've seen before, that nicely fits into the chip well, into the 96 well of the tissues chip. And then we have everything already, right? We don't have everything built up. Of course, the cells are not well characterized, but for some questions, it's not important yet what the immune cell composition or the ratio is. Maybe you just want to see, in this case, Martin was approached by another institute and they had this new material and they just wanted to know, hey, Martin, is this really bad for bone? And so Martin was like, give me a couple or maybe 4 to 6 weeks and I will see. So this is what happened. Martin used the control where he just drilled a little hole. And I just want to say that like all of those things, they're not like no company is helping you doing these kind of things. So Martin had to be super creative how to drill a tiny hole into a tiny bony piece and then inserting this tiny new material into it. But he did that and he monitored again LDH, so the cytotoxicity. And what is nice that Martin could, or we could actually report that this new idea was not on the first look that bad for the bone. And we were also able to do CT on that. Here you can see bone formation and bone resorption. Again, if you have a lot of bone loss, that's bad. But here, Martin could actually observe that this nail was not influencing a bone homeostasis in a bad way. Another readout parameter that I would like to briefly introduce is called CTX. It's an ELISA that you can do. It's a metabolite that is present most of the time, but if it's excessively present, that means you have a lot of bone resorption because it's a very exclusive parameter that you can measure when osteoclasts are working away, but not new bone is formed. So Martin was curious, is a lot of CTX released? Does it mean we have some unwanted bone loss? And actually, the ELISAs that he did so far showed that there was no significant difference between with nail and without nail. So LDH can actually go a long way. Sometimes it's just nice if something's cytotoxic, because if you are an institute that just wants to develop a new material and you're an engineer, you just want to know if I'm on the right track. So Martin also looked over time and could also say that LDH is not really going up way high and indicating some cytotoxicity derived from the material. So he could report back to his colleagues that they could go on with their idea of this new material. Another thing was just to show that we are thinking in different direction and what's possible. I told you that we use a lot of the synchrotron radiation, which is X-ray. So we were also curious, how is this transporting the chip? Because actually, if you do CT or if you do the synchrotron, you have to transport the samples as well. And we just wanted to see whether that's affecting the system. And so this is just nice to see that one essay and one good idea can answer a lot of first research questions. Next, I told you that we have the osteoblasts and the mesenchymal stromal cells, and those are cells important for the bone. And I also told you that we can isolate them, or my colleagues isolate them from primary tissue. The real problem is that osteoclasts, the ones that resorb bone, that they are actually not, you cannot isolate them from native tissue. So we actually had a really bright master student joining our group. Now she's a PhD student, or you shouldn't say student, she is a scientist in our group, and she has been showing us how to differentiate osteoclasts. I said before, you need monocytes, which then differentiate to macrophages, which then fuse and then form the mature osteoclasts. And actually, this is quite a hassle. So it's not that easy. So when I told you before that I was hoping that I would just have osteoclasts on my system, I didn't. I never had osteoclasts on there until Nina joined the group. And the thing is that you have to differentiate those cells directly on bone, on the bone scaffold, because once those cells have fused, you cannot get them off anything. Nina has, with another colleague, they have been trying to scrape off those cells from tissue plate, and it was just, according to her, it was quite a ride. So you do have to differentiate osteoclasts on bone directly. But when you do this kind of work, a lot of new questions arise. So actually, Nina came up with a lot of different questions when she was working with the 3D environment and the chip and the osteoclasts. Like, for example, how many monocytes do we need? Because they differentiate into osteoclasts later on, right? Which cell type do we seed first? How does media influence all of this? And how do other cells, like the bone-forming cells, the osteoblasts, and the mesenchymal stromal cells, how do they play into this whole thing? And then what is the better source of monocytes? Because I showed you that they're in the blood, in the bone marrow, and you actually have stem cells. For people that are not familiar, or maybe not biologists, there are some special stem cells that you can alter. Maybe that's too. Yeah, but basically, so you can differentiate osteoclasts from all three sources. So Nina was wondering if all of this is working equally well. And actually, yes, it does. And it really depends on your research question and also on what's available. Because raising iPSCs, those stem cells I told you about, they're actually really hard to culture and to keep in check. But when you have blood, like just regular blood, I mean, it's quite common then that in a clinic blood is drawn. So you have access. You can actually order blood. This sounds weird for people that don't do it, but yeah, it's a thing. So blood is usually available. When it comes to bone marrow, you actually have to have access to bone marrow, which a lot of labs don't. So Nina showed that it's possible from all sources, but which one you use depends on the question you have later on. She then was wondering which cell type do we seed first? And actually, again, proving that I never had osteoclasts on my first bone marrow chip is that MSCs and osteoblasts, so the mesenchymal stromal cells and the osteoblasts, they actually inhibit the differentiation of monocytes to osteoclasts. So if you add them together, they will just stop the process and you will not have the osteoclasts on there. And she did that with what we call a 2D plus co-culture. This is also a nice way to just show cell-to-cell interaction, and osteoclasts need bone, right? So she added little bone chips. This was a pretty nice experiment, and now we kind of know a little bit better in what sequence we do have to start our chip. Other readout parameters that Nina is currently assessing just for her questions is the bone cell activity, bone formation, and resorption. Here you can see again the CT, really tiny CT, super cool. And you can see bone formation and bone resorption basically in balance, because you don't want one cell type to overshoot, making a lot of bone, or the other one resorbing a lot of bone, right? Because that would be pathological. Also, immunohistochemistry staining for osteoclast or matrix is a nice way to just visualize if stuff's happening and if it's going in a wrong, right, not wrong, maybe also wrong, but right direction. And then again, you heard this before, the CTX to show if bone is lost, LDH, if cells are dying, and other markers to see if bone's formed. So I promised you immune cells and microenvironment. So this will be now moving towards the end. It is actually really hard. Immune cells are really tricky to culture altogether. We have seen studies where you have a lot of T cells, or maybe where you can maintain B cells, but having the whole picture of bone marrow is actually pretty hard. So I'm just showing you one attempt of maintaining that. And here you can see the big subsets of immune cells. So those are all the cells that make you survive any kind of viral, bacterial, or whatever infection. And we do maintain T cells and B cells and monocytes and all of them over time. But of course, I cannot fool you. You see that there's a lot of change. So of course, we have cells dying. But what was really cool was that B cells were actually surviving without us doing anything, right? If you're a B cell biologist, you know that you have really give them a lot of attention in order to survive in a 2D culture. But we were very surprised to see that our little B cells are pretty happy over a pretty stable time. But of course, we lose them at some point. So, but this was a cool assessment. And also looking at the microenvironment, the nice thing about the chip is that you can always take a little bit of supernatant and then do an ELISA or whatever when you do a media change anyways. And we could actually see in our cytokine array that we had OPN and OPG. Those are just important peptides for bone formation, that they were actually there the whole time. So this was just nice. And just to show you with this really nice heat map that we did a lot of things, is basically just to illustrate that you have to start somewhere. So you have a lot of different cells, you have a lot of different media, you have time going by, you know, so we don't know much about human bone marrow, to be precise, so, and to be honest. So we just try to see what's in there, and we try to make educated guesses, but in the end, you end up just doing an unbiased screening and then see what's interesting and just follow up with that. So I have been doing a lot of those cytokine arrays, and it's still really cool to see what's happening. And I really want to point out lots of cytokines that we say are, for example, pro-inflammatory, so are part of a cascade when you're sick or when you injured yourself. They're not pro-inflammatory in the bone marrow. They seem to be important for the development of some cells. So this is another thing where I also think focusing on especially murine, so mouse bone marrow, has been misleading, because when we look into human bone marrow, we find a lot of interesting stuff that would be characterized as pathological, but it seems to be important for cells to actually go into the next development and differentiation step. So I think we need to really rethink human bone marrow. Yeah. So now coming to our hopes and dreams, I would really like to call it that, because some of it probably will never work, and some of it probably will work. And now the promising ones. Again, my colleague Annie from earlier before, she's actually part of a clinical study where we test drug in people that broke their arm, which had a fracture. And Annie is actually trying to rebuild this whole environment on our bone chip and wants to do that parallel to our ongoing clinical study, which is pretty nice, because if you have inflammation, it's not really that you have to have the whole thing that's happening in a fracture, but if you have some sort of injury and a response and inflammation, and you can then see an anti-inflammatory shift, that could maybe answer some questions what's happening in the patients. And if you then remember how it worked with the metal on our chip, Annie would just go back and forth between the clinics. So it's basically like, is this what it looks like in patients? And then they say yes or no. So she goes back to the lab, and, you know, so, and sometimes we find things, and then we ask clinicians, we're like, have you seen this before? And they're like, no, but it's interesting. So it's really nice to always go back and forth and actually talk to patients, talk to doctors, talk to clinical staff, and ask them, is this really happening, what we see in the lab? And Annie is very involved in this, and maybe she's presenting that work in some time ahead of now. Another one is Nina, the person with the osteoclasts. Nina actually started because of a very rare but severe disease called osteopetrosis. There are several forms of osteopetrosis, but the one she's looking into is actually pretty bad. So most of the kids die at a young age because they make too much bone. So the osteoclasts, the bone resorbing cells, are not removing the excessive bone. So the osteoblasts just work away, and the osteoclasts are basically sitting there, not being able to do their job, and then basically your whole body becomes bone. And for this, there is an idea for a gene therapy, but of course, this is a very individual disease. So Nina would like to do a safety assessment on the bone chip. So in this case, she would use the iPSCs that I showed before, differentiate the osteoclasts, and put them on the bone chip. And that's the easiest way in order to test a gene therapy, because you can just alter. iPSCs are the ones where you can make modifications. So this is another interesting way, because in the bone chip, she could also go back and forth and see if what she's altering, is that also the same mutation or the same change in her osteoclasts that she sees from patients. About my future and my hopes and dreams, I really feel this comic because I have attempted science, and I had this idea, and I wasn't really wrong, but I really love this comic. Because it says I was wrong in numerous ways, and then this poster reviewer says there are numerous diagrams. Well, I produced a detailed tribute to my wrongness. I also produced a lot of data, and my conclusion is that bone marrow is heterogeneous. So now you have seen a lot of the bone, a little bit of the bone marrow, but when I worked with the chip, I realized that we don't know enough about human bone marrow. We think we know a lot, but we don't. So every time I ask a question, hey, have you seen this in human bone marrow? They're like, we don't go that deep into that tissue. So I actually went back, and it could fill another whole talk. I actually went back to the clinic, asking my colleagues for more bone marrow samples, working with the core unit, trying to figure out what is natural heterogeneity between patients and what is an observation that I have in vitro or in my 2D or 3D culture. Because many times I know that most of you maybe work in this field, and you know that we have to do a lot of work in a very short time with very little money. And sometimes it's hard to go back to the roots, but I really had to go back to the roots, because as of now, I don't know yet. I don't know what human bone marrow looks like. And if I don't know, then it's hard for me to assess that on the chip. We do have some ideas, and we do keep some major subsets of cells alive, which is nice. But when you go deep into like the subsets of, for example, B cells, then it's actually quite hard. But it's fun. But this was not the scope of this talk. So yeah, just to let you know, maybe I will be back with a bone marrow talk. And yeah, moving on to the summary. Hopefully today you learned or somewhat understood that osteoblasts and mesenchymal stromal cells can be isolated from biopsies that we get from surgeries, but osteoclasts cannot. That bone marrow is heterogeneous, especially the immune cells. That when you seed your bone marrow chip or your bone chip, that the order that you seed the cells in and the right ratios that actually matter, they're of importance, because you learn from Nina that if you add osteoblasts and MSCs prior, your osteoclasts will not differentiate. That soluble environment and cytotoxicity can be monitored over time while your chip is running, which is nice, because then you don't have to arrest your experiment in order to assess that. You can just take a little media, do your assay, and let your chip do its thing. You also learned that, and what Martin did, that native bone marrow can be easier to handle and is sufficient for basal research question, especially if you just want to know, is this the right way or not? Then with Annie's experiment and her clinical study, you saw that it's important, and also with the metal implant research question, it's important to benchmark the system to the closest reality available. For us, that's the patient. I'm a very strong advocate for benchmarking things that are human to humans and not to other species, because it doesn't make sense. I'm sorry for everyone who thought that, but it just doesn't make sense. I have very strong feelings about that. And then again, primary material can be diverse. And also for maybe the early career scientists here or students, it is okay if it's not working. Like, it's biology. It's not supposed to work all of the time. If it works 100%, it's not biology. It's something else. But sometimes cells don't work. Sometimes your patient has some condition they don't know about. Sometimes it's okay. Maybe when you're older or when you do this and that, some cells don't make bone anymore, and maybe that person still lives a happy and healthy life. So if your cells don't differentiate, it's okay. Just always quality check. It's really a waste of time if you don't do the extra step, go all the way in 3D, and after 42 days you realize, oh, they never lived a good and healthy life, those cells in my chip. So do pre-testing, go the extra mile, and then have a sufficient system to answer your question. And sometimes it doesn't even answer your question, but that's just the thing with organ-on-a-chip technology. We're still kind of like in kindergarten when it comes to those things. So I really hope that we get more time and understanding also from funders, that we have a lot to learn, a lot of pitfalls. And it was really good to have the money from Kat and from Berlin, because with that we could actually do a lot of things that we couldn't without. So with that, I would like to thank the Berlin State and Kat Herman for her funding, and the whole group. This is everyone. We're not always as happy because it's a struggle to work with human samples, but we're pretty happy when it comes down to it. So thanks everyone, and I would really invite you to ask questions